Welcome back, class. Mr. Rasmussen here. We've been talking about cellular respiration. So part 2 is going to cover just glycolysis. We said in the last lesson that cellular respiration contains three main stages: glycolysis, the Krebs cycle, and the electron transport chain. Well, in this lesson, in part 2, we're just going to talk about glycolysis. Now, glycolysis, the name glycolysis, has two parts to it. It has, as soon as I find my pen tool, there it is. Over here, it's got glyco, which is one part, and lysis is the other part. Well, lysis, we've seen this. We saw it in the phrase hydrolysis, and lysis means to split something. And glyco is a prefix that means essentially sugar. So the whole idea of glycolysis is going to be breaking sugar in half or splitting sugar. So that's what we're going to talk about here. It's what happens to sugar, how it gets split, breaking apart sugar. So at the end of each of these, you'll see a chart with all the information that you need to know, all the inputs and outputs of each part. And then I want you to have a good idea of each stage, a good idea of what happens. So, step 1, glycolysis. Glycolysis takes place in the cytoplasm of the cell. Cells don't need mitochondria to do glycolysis. So if we're talking about a prokaryotic cell, a cell that doesn't have mitochondria, that lacks those organelles, they're only going to be able to do glycolysis to gain their energy. They're going to attach something to the end of it to get rid of some of these molecules, but they are only going to be able to do glycolysis to get their energy. So it takes place in the cytoplasm, does not require mitochondria. And the main thing we're going to do is we're going to take glucose and we're going to turn it into this molecule called pyruvate. Pyruvate. And when we do that, we're going to generate just a little bit of energy. We don't actually generate a lot of energy, way less than the electron transport chain, but we're going to generate some energy. So glucose here has 6 carbons, and pyruvate has 3 carbons, so it's a 3-carbon molecule. So essentially what we're doing is we're breaking glucose in half. We're breaking glucose in half. And while doing that, we generate some ATP. Here's the picture that we're going to see a lot of, and I'll talk through it in the next slide. So here's the picture that you're going to see all the time. We're going to take glucose and we're going to change it to all these little intermediary molecules, and we'll go from there. But we'll talk about that next time, or in the next slide. So glycolysis requires an input of ATP. So it requires an input of 2 ATP. So in order to make glycolysis actually happen, we have to put in a little energy, and I'll show you that. Right over here, we start with our glucose molecule. So here's step number 1. We start with glucose, and we have to put in ATP. So here's ATP, and here's ATP. And that's ATP going into the process. So we had to put 2 ATP into it in order to make this process work. So we go from glucose, we put an ATP into it, we get this phosphorylated, right? So that phosphorus from the ATP. So we get glucose 6-phosphate, and then that changed to fructose, and then we phosphorylate it again, or we add energy again, we energize it, and now it has these 2 phosphoruses on it. Okay, and then it splits. It breaks apart here. We split the molecule, and here's where we start getting energy out of it. So now after it splits, this process over here, you can see this whole thing, it happens twice. So we have this process. So what happens is we get this process and we get all these intermediary molecules, and after we get these intermediary molecules, we start to make some ATP. So we make ATP here, and then we make ATP here, and then we do that whole thing over again. We make ATP again, we make ATP again, and in the end we're left with this molecule. We're left with pyruvate. And we're not left with one molecule of pyruvate. You can see the carbons are right here. These little gray dots, carbon, 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 3 of them. 1, 2, 3 carbons. Okay, so pyruvate has 3 carbons. It's half of a sugar molecule. And we're not left with just 1 pyruvate, but we end with 2. So we split glucose in half. So what we do, first we take glucose, we energize it. So step 1 is we start with glucose. Step 2 would be we energize it with ATP. Okay? Step 3 is it splits. Okay, glucose splits. And then step 4 is we pull some energy out of it. We harvest some energy out of it. And we actually get some NADH as well. We get some NADH, this high energy carrier molecule, right down here in the bottom. So we make some NADH as well. So we made 2 ATP and 1 NADH, and we do that twice. So we make a total of 4 ATP and 2 NADH, but we had to spend 2 ATP. Does that make sense? So we had an input of 2 ATP. 2 ATP went in. So we're basically minus 2 ATP, but we gained 4 ATP. So which means we have a net of 2 ATP created. A net of 2 ATP created. Okay. So it also produces some NADH. I wanted to put up a chart for each of these so that you know what goes in and what comes out, the inputs and outputs of each of these processes. So glycolysis, the input is 1. We're beginning in 5, 4, 3, 2, 1. I created a chart for you. In this chart, I wanted to give you the inputs and outputs of this process, so what goes in and what comes out of each stage of cellular respiration. And so our inputs are 1 glucose goes in. We get a couple of NAD+, which gets turned into NADH. We get 2 ADP, that eventually gets turned into ATP. And we also have an input of 2 ATP, so we had to spend some energy. Our outputs are that glucose becomes 2 pyruvate molecules. The NAD+ gets energized and becomes 2 NADH molecules. And the ADP becomes ATP. So we get a total of 2 net ATP produced. So those are our inputs and outputs. Now I want to talk a little bit more. There's a little bit more to this story, and we added on before we get to the Krebs cycle. It's called the transition reaction. The transition reaction. So something happens before the Krebs cycle. After glycolysis, we end up with pyruvate, but then something happens before that molecule actually enters the mitochondria, and we go to the Krebs cycle. And here's what happens. The transition reaction takes place in the cytoplasm. It connects glycolysis to the Krebs cycle, so it's going to connect these 2 stages. We don't actually call it a stage, it's like a stage and a half, so like 1.5 stage. And here's what happens. So we ended up with that pyruvate molecule in glycolysis. So our ending process of pyruvate, our ending molecule of glycolysis was pyruvate. That pyruvate in this transition reaction gets turned into this thing called acetyl coenzyme A. Acetyl coenzyme A. So pyruvate gets changed into what we call acetyl coenzyme A, or acetyl CoA, you'll hear it called. And when that happens, we make something. We make a little carbon dioxide. So there's a little carbon dioxide given off as waste. A little bit. We also make 1 NADH. So we make a little bit of this, one of these energy carrier molecules. So we make 1 of those. But that's not the whole story, because there are 2 pyruvate molecules at the end of glycolysis, so which means this reaction happens actually twice. It happens twice. So because of that, we get 2 of these produced total. 2 NADHs produced total, because this whole process happens twice. So the transition reaction, we take 2 pyruvate, we convert it to acetyl CoA, and we generate 2 NADH. We get 1 NADH each time a pyruvate becomes an acetyl-CoA. Pretty simple. That's a transition reaction. We go from pyruvate to acetyl-CoA. We generate 2 NADH. So here's your summary to that. Our inputs are 2 pyruvate. Those become 2 acetyl-CoAs. We have to put 2 NADHs, NAD pluses, because they become 2 NADHs. And then we have 2 COAs that go in there as well, and 2 carbon dioxide that leave. So those are our transition reaction summary. This has been lesson 7B, and yeah, if you have any questions, please ask me in class. Have a good day, team.